Hi there and welcome to this IB revision video with Mr. Phillips, and today we're looking at biological membranes and the movement across those, and this is the IB course from 2023 onwards for higher and standard level. And please remember, this will support you but it's not the complete revision process. So this is an old question, but I think it's one worth you seeing, especially if you've just watched the video on eukaryotic cells and organelles, because there are membranes within cells as well as the cell plasma membrane. So just be aware that when you are being asked about membranes, that it could be asking you about the cell surface membrane or the plasma membrane, or internal membranes such as the crystallini, for example in mitochondria. So just so you're aware, if we're looking at functions of the plasma membrane being the cell surface membrane, the outer membrane of the cell, and obviously that isolates the cell from its surroundings and it permeates what materials can enter and leave the cell. So it permits what can enter and leave the cell. All living cells have a membrane, so it's a key thing. Every single cell has a membrane, okay, and it controls the exchange of materials between the environments of the cell. And membranes also have need to have an ability to receive messages, so they'll have receptors, which are often proteins or glycoproteins, to receive messages from chemicals such as hormones entering the cell. Within the cell, as we've spoke about in the eukaryotic cells video, it allows compartmentalization, so separating sections of the cells apart so they can do different jobs. So for example, the membranes that make up the mitochondria allow respiration. In the same cell, a plant cell can be doing photosynthesis in the chloroplast. Also, you can hold membranes in a certain place, hold enzymes within the membrane, which might allow a series of enzyme reactions to happen in the right order. It presents disruption in the cell, so lysosomes, for example, may be used to break down old cell parts. It allows vesicles to form, as in the Golgi apparatus, in the trans side of that, and it allows exocytosis when the vesicles fuse to remove a substance from the cell. And hopefully from previous video you will be aware that there is phospholipids are the main structure, and you should be aware that we have this phosphate head which is hydrophilic, okay, so that's represented by this bit here, and then two hydrophobic tails, fatty acid tails, which are here, you can see there. Remember, there's only two as opposed to triglyceride which has three, and then ester bonds bond those together. Now we think the first phospholipids made in the first cells, which we'll talk about in the next video, would have come together into something called a micelle, and then over time they've evolved to become a phospholipid bilayer. And the bilayer is formed when surrounded by water. It can move like a fluid in the plane of the membrane, and sometimes, so what this means is that this phospholipid can move over here, this will move over here, and sometimes they'll even flip-flop opposite each other. They're approximately 7 to 10 nanometers thick from here to here, and because they have a hydrophilic head pulling outwards and a hydrophobic tail pulling inwards, this makes the membrane quite stable, even though the membrane molecules are not bound. The membrane is partially permeable, which we'll come back to a bit later, which means molecules can get through there, and particular water molecules can get through, which we'll talk about through channel proteins. Some membranes have higher permeability due to these water channels and aquaporins. So then, what do we need to know? We need to know that the model that we use, it's a model because it's a simplified replication of the cell membrane, is the 1972 Singer and Nicholson fluid mosaic model. And this is the idea that phospholipids are freely floating within the bilayer, and within there are proteins that are sticking out called extrinsic. and ones that span the whole layer called intrinsic. So this channel protein here would be intrinsic, this protein here would be extrinsic, because it's only on the outer edge of the membrane. And here you have the different parts, the different things that we have. So we've got actin myosin here, which are involved in movement. We've got cytoplasm inside the cell. We've got phospholipid bilayer, cholesterol, glycoproteins, and glycolipids. And we'll talk about their role in a few moments. So the membrane structure, as I said, is described as a fluid mosaic model. Why is it described as that? Well, the term fluid links to the phospholipids because they are free to move within the layer of the membrane, and the mosaic model is the proteins because they look like a collage of different proteins stuck together if you look at it under a microscope. So you need to know the different features and the roles of the different features of the fluid mosaic model. So you need to know that it is a phospholipid bilayer. You need to know that the phospholipids are free to move within their own layer. You need to know that the heads of the phospholipid, which is the phosphate head, is hydrophilic, whereas the fatty acid tails are hydrophobic. You need to know that it has unsaturated phospholipids and also can have saturated phospholipids. And the more unsaturated phospholipids, which we'll talk about in a moment, the more fluid the membrane is. Cholesterol will make the membrane more stable, and cholesterol stops the membrane breaking apart. And in this diagram there isn't really any cholesterol, so just to sort of show you, the cholesterol, as we've talked about in a previous video, is 4 carbon rings, so it would appear between the phosphates, the phospholipid tails in the membrane. Membranes become less fluid at low temperatures. Most proteins are free to float about, but some proteins are anchored in by internal structures such as actin. Some proteins span the membrane and are held in place by the hydrophobic interactions that they have with the hydrophobic tails. Some proteins are joined to carbohydrates forming glycoproteins, and some lipids are joined to carbohydrates forming glycolipids. So let's have a look at some of the roles of these key things. So first of all, the phospholipids act as a barrier to most water soluble substances due to the fact that we have this hydrophobic layer on the inside, or the tail here of both sides is hydrophobic, so water cannot freely pass through the molecule. We have cholesterol, as I said, which is a 4-ringed carbon structure, and it regulates the fluidity of the membrane. It's by allowing to give more gaps between the phospholipids. It stabilizes the membrane though by keeping them stuck together, and it can prevent ion and polar molecules from passing through the membrane. Proteins provide hydrophilic channel proteins for ions and polar molecules to pass through. Proteins may also be enzymes such as ATP synthase, and glycolipids and glycoproteins can also stabilize the membrane structure, but the main thing you'll talk about them doing in this course later on is acting as receptor molecules for cell recognition, and also within bacteria, where they therefore become the antigens that our body recognizes, our white blood cells recognize as being non-native to the body. And they will also form a layer, and this is quite important, hence it's in bold. They'll form a layer on the outer membrane called a glycolax, and the glycolax then allows the fusing of cells together. So they're an outer layer, and it fuses, holds the two cells together to make a tissue. So remember, a tissue is a group of cells working together for the same function. Now, one thing you need to know about is membrane permeability, and what you should be able to do is look at a graph like this and understand how permeability changes with temperature. So what this is showing is if this line here is 0 degrees Celsius, if it's lower than 0 degrees, the colder it gets, the more permeable, the more things can pass through the membrane. That then gets lower to around 10 degrees where it's about the lowest, and then that increases again towards higher temperatures. Now we need to understand why, so let's look at A first. Below 0 degrees, phospholipids have very little energy, so they don't move very much. They're packed closely together like solid, and the proteins denature. Although I don't really like that word if I'm honest, so I would say the proteins don't work as well, and they can lose the structure and function. to allow some particles through. One reason this may happen is because ice crystals may damage the proteins and pierce the membrane, meaning it becomes highly permeable when that membrane thaws. However, between 0 and 45 degrees, you're adding more energy to the phospholipids, so therefore they will start moving. But there'll be very little protein denaturation at that temperature, because that's the optimum range of temperatures for proteins, so very little leakage. So as the temperature goes up here along here, if you watch the graph, you're getting more and more energy, phospholipids vibrating more, meaning more gaps appear, so things can move through the gaps between the phospholipids. And above 45 degrees, at this temperature, the phospholipid bilayer starts to break down. The water in the cell may expand, which causes pressure on the membrane, and that means that the water may want to actually want to leave the cell, so the water sort of inwards pressure out on the membrane, and the channel proteins and carrier proteins will denature, meaning they no longer control what enters and leaves the cell, and they may change shape, leaving gaps between the cell. Now, another thing that can disrupt a membrane is the use of solvents such as acetone or ethanol, which can damage or dissolve the lipids. So lipids will dissolve in ethanol, for example, and therefore that would cause the dissolving of the cell membrane. So now what we're going to look at is section B, membrane and movements across the cell membrane. So the first term we need to use is simple diffusion. So simple diffusion is the net movement of molecules from an area of higher concentration or a region of lower concentration, which can also be described as down a concentration gradient. Now be very careful with this in exams, because a lot of people understand this but miss the marks, and that's because they don't say higher, so this ER is important, lower. And the reason that's important is if you've got a scale, so let's say we had 5, 4, 3, 2, 1, and over here the concentration, let's say, was 1. Okay, it would still move from 2 to 1 because 2 is higher than 1, and therefore even though 2 is quite low, it is higher and lower. So comparative language is really important. The way to avoid that is saying move down a concentration gradient. Always, however, say the word down, do not say the word along. This then occurs, basically the reason it's called simple diffusion is particles are passing directly through the phospholipid bilayer. So like this molecule here moves through the gaps between the phospholipids and therefore is called simple diffusion. Now, things that might affect this is the steepness of the concentration gradient. So obviously this 2 to 1 that I talked about here, if it was 5 to 1, that would be a faster rate of diffusion because there's a higher difference between the two. The temperature affects the rate of diffusion, especially if it's going through a membrane, because the membrane may be more permeable at higher temperatures, but the particles diffusion will also have higher energy and move faster as a result. The surface area it's occurring over, so hence the alveoli have a large surface area to increase the rate of diffusion, and the properties of the substance's diffusion will obviously aid diffusion. So smaller molecules will diffuse faster than bigger molecules, lighter molecules will diffuse faster than molecules of a larger mass. So what substances can do simple diffusion? Well, the first substance is oxygen. Oxygen is non-polar, so it's not affected by the hydrophobic nature of the phospholipid tails. It needs to be able to diffuse straight across the membrane, because otherwise we would struggle getting oxygen into our cells. Likewise, carbon dioxide is. Now, carbon dioxide is slightly polar, but it is small enough to pass through the membranes quickly. So again, I always think if you remember that those two molecules are essential for life, then those two molecules can diffuse through and must diffuse through quickly. Water is polar, but again small enough to diffuse through, but we do have aquaporids to support it in some areas, which are basically channel proteins that allow water movement. And lipid molecules, anything made of lipids can, because they are, the membranes are lipid soluble, so they'll fuse, the lipids will fuse into the membrane and through the membrane, and this is how steroid hormones pass across. Now the key thing is that the centre of the membrane is hydrophobic, and what that means is that ions, so things with a charge, cannot pass easily through as they will be repelled. So that is something we need to look at in a little bit more detail. So substances that can't pass through just by a simple diffusion is glucose and amino acids, because they're too large, so they use something called carrier proteins, and as I said a moment ago, charged ions or polar ions, which are charged and therefore get repelled by the different layers in the membrane, so they have to use channel proteins. This process is facilitated diffusion. Now it's still called diffusion because it's still from a high to low and is still passive. However, they use these molecules in the membrane and their natural movements to allow them to move from one side of the membrane to another. So to support with the movements into membranes using facilitated diffusion or active transport, there are lots of proteins within the cell membrane. Now there are two groups of proteins. So the first group is what we call peripheral proteins, and peripheral proteins exist just on the outside of the outer layer of the membrane, and are just embedded just inside the membrane, as you can see here. Some are anchored down by a single hydrocarbon chain, and they may attach temporarily to integral proteins as well. But their role might be that they are hydrophilic, which means they might then become enzymes within the membrane. Integral proteins, such as this one here, which is clearly a channel protein, have a surface that is hydrophobic, and therefore that's this bit here, so that will embed then into the hydrocarbon chains in the membrane. So this outer surface here is hydrophobic and the inner surface here is hydrophilic, which allows then materials to move through the transmembrane channel and allows the movement of particles via facilitated diffusion, such as charged particles. Now, what type of proteins there are in the membrane will depend on the role of the membrane. So membranes obviously have an inner surface and an outer surface, okay, and they'll have their protein orientation will depend on the role of the protein. But generally the idea is the more active cells tend to have membranes which more integral membranes across the whole membrane, because they have to get more materials in and out of the cell. And some of these membranes have specific shapes. So here we can see a carrier protein, and you often see this version here, this 2D version of the glucose entering the cell, and then the carrier protein changing shape, and then the glucose going into the cytoplasm. Well, in this case you can see that that is the case, but the glucose molecule has to fit perfectly into here to allow that to happen. Now there are two main protein types, as I said, two main integral protein types, and they are channel proteins and carrier proteins. So ions and polar molecules can't pass through the phospholipid bilayer. We've said that because if they were trying to do so, they would go for here and either be repelled by the positive head or the negative tail. So they have to have channel proteins because they have a hydrophilic internal structure, so water can travel through it, and that anything dissolved in that water can go through here as well. The channel carrier proteins then allow molecules to go in and again change shape using just the general movement, natural movement of the membrane to not use energy and just facilitate that diffusion. That's for large molecules such as glucose. Most solutes, therefore, most things that dissolve will travel through the membrane through proteins, and it is diffusion as it doesn't involve energy. So each protein channel is specific and it will allow certain things to go through only. Carrier proteins, we've said, these will change shape, and it's called sometimes the ping pong or the flip-flop mechanism as a result, allowing the material through. Again, this is passive because it is facilitated diffusion and it is going with the diffusion gradient, down the diffusion gradient from a higher to a lower concentration. However, at some points we need to use active transport. So active transport is obviously when we're going against the diffusion gradient from a lower concentration to a higher concentration. So simple diffusion goes straight through the phospholipid bilayer. Facilitated diffusion uses either carrier proteins or channel proteins to go down the concentration gradient from a higher concentration to lower, but through proteins facilitating it, whereas the active transport uses ATP to force molecules against the concentration gradient from a lower concentration to a higher concentration. It differs from channels proteins that we've talked about before because it requires energy, only allows the movement in one direction, so whereas these ones, the molecules may move in both directions, here this is only happening upwards. So when it's active transport, facilitated diffusion, it can go in both directions. When it's active transport, this mechanism only works in one direction as designed for that membrane. And normally it's going in a pump protein across, and normally against the concentration gradient across the membrane. Now the key thing about this is how does it work? Well, the protein pump basically moves from one shape or one conformation to another one. So as you can see here, what happens is the molecule coming from here, remember from the lower concentration to the higher concentration enters, it will have a specific shape which will fit into the carrier protein, and the molecule enters from the low concentration and binds to the complementary shape. ATP is then used to release energy that changes that shape, so that ATP releases energy to a second conformational shape, which then releases that molecule into the higher concentration as a result. So when we look at water, and water in particular, we are looking at osmosis. So water often moves across boundaries, across membranes from osmosis. So osmosis is the diffusion of water from a higher water concentration, where there's lots of water, it's dilute, to a lower water concentration, where something is more concentrated, through a selectively or partially permeable membrane. And the reason this can happen, the reason water will move through, is if you imagine this water molecule here, if water is pure, it can easily move and fit through the gaps in the membrane. If, however, water has got a dissolved substance in it, so let's say this, we've got the water molecule there, but now it's dissolved the substance, so let's say it's glucose is dissolved in it. The water molecule will have glucose sort of surrounding it like this. It will be then too big to move through this membrane here. So if you've got lots of water one side, it will tend to move through the membrane to the other side because it is free to move through, whereas the dissolved solution isn't as much. Water can move through all membranes, even though it's polar, because it's small enough to pass through the phospholipid bilayer. Now, some cells are adapted to aid osmosis, and these would be using specialized channels called aquaporins. Think about the word aqua means water, porins means hole or pore, and therefore water is able to move through the channel. Now, water is able to move through the channel into cells, so often very common with stuff like kidney cells and things like this. Now, these cells are adapted, these proteins are adapted, I should say, to control what moves through. So they have a very narrow point, so you can see that sort of here, okay, where it's only wide enough for one water molecule at a time to travel through, and that is useful because it stops other molecules getting through. Now, water is a very small molecule, H2O, so it's got a relative atomic mass of 18 only. Positive charges such as hydrogen are repelled, so if there's any dissolved substances in here with a positive charge, so let's imagine there's some hydrogen floating down here, that hydrogen charge will come down and it will be repelled back out by the positive charges on the protein. There are therefore lots of ways that molecules move through the membrane, and you need to know the different methods and be able to recall them and explain why they may or may not happen in different scenarios. So you really do need to ensure you explain what simple diffusion is. Anytime you mention simple diffusion, you need to say it's going through the phospholipid bilayer, not through the proteins. You need to be able to talk about facilitated diffusion using channel proteins and carrier proteins and not using energy because they go from a higher concentration to a lower concentration. And the channel proteins, you do it for charged particles as they have a hydrophilic center. Okay, whereas the carrier protein is for larger molecules of specific shapes. And active transport is using energy, going from a first conformational shape to a second, using ATP to change that shape and push the molecules across the membrane against the diffusion gradient from a lower to a higher concentration, and it uses energy to do so. So section C then is going to look at what can affect the fluidity of a membrane. And the fluidity of a membrane means basically what materials can pass through it and how much the molecules move within the membrane. So first of all, this all links to the composition of fatty acids and the phospholipids. So there are two groups of phospholipids. So this first group is saturated, and what saturated means is that there are only single bonds between the carbon atoms. So there's no double bonds whatsoever, and that means that every bond between those carbon atoms is used up, so you can't add any extra hydrogens in at a later date. Whereas unsaturated means that somewhere, and it can be more than one, this is be monounsaturated, you have a double bond. Now what this means is it's unsaturated, because in theory you could break that double bond and add some more hydrogens in there. But the important bit is what happens to the shape. The saturated fatty acids are pretty much a straight line, and if you look over here at picture B, you can see here that the phospholipid tails as a result are pretty much straight. What that means is that the phospholipids can be very close to each other and therefore act as more of a solid material, just vibrating in position. However, when it's unsaturated, the fatty acid tail has this kink in it, and this kink means that the molecules cannot be as close together. So the more of these unsaturated fatty acid tails we have, the less viscous the membrane is, the more fluid it is, because the molecules of phospholipids cannot get as close together. And therefore additional materials might be able to get through. And this is important because membranes must be fluid enough to allow materials to enter, but be strong enough to not be perforated and not allow anything to enter. And there's an exact ratio that will be dependent upon the temperature. So if we look at this, Arctic fish have a higher percentage of unsaturated fatty acids. So what that means is Arctic fish have a higher percentage chance, have a higher amount of fatty acids that will look like this. And the reason for this is at low temperatures, the phospholipids will vibrate less. And if you had phospholipids that are more like box B, they might freeze together and make the membrane completely impermeable, completely unfluid, and kill the cells. Whereas in box A, they're more fluid, so even at really low temperatures, the phospholipids are unlikely to stick together and sort of act as a solid. Now, cholesterol, as we've talked about previously, is important in this as well. So cholesterol is not the same as a normal lipid. It is based as a lipid, but it's made of 4 carbon rings, as you can see in this diagram, and it basically sits between the phospholipid tails, regulating fluidity and strength, and it makes approximately 20 to 40% of eukaryotic cell membranes. Again, different amounts depending on the conditions that that organism is living in. Most of the molecule is hydrophobic, so hence it sits in the tails and it is attracted towards the hydrophobic tails. So it's pulling towards the hydrophobic tails of the phospholipid. But on the end, its hydroxyl group is attracted towards the phosphate head. So that bit is going to be pulled towards the phosphate head. Okay, and the role of it is at high temperatures it stabilizes the membrane, okay, because it will stick between the phospholipid tails and allow them and prevent them from coming apart, and acts as like glue almost between the two of them. Okay, and it also and that's particularly important, I should say, in in unsaturated fatty acids. It will also prevent saturated fatty acids from solidifying at low temperatures. So it has two roles. At high temperatures, it maintains the strength of the membrane and maintains its impermeability so it doesn't get too fluid, but at low temperatures it prevents the saturated fatty acids from sticking together and preventing stiffening of the cell. So it really does regulate the fluidity of the membrane. You also just need to be aware of vesicles, and we've talked about this before, so I won't spend long on it. You need to know a vesicle is a sac of membrane, basically, and you can see here how a vesicle forms during endocytosis. So the vesicle is an infolding of the cell plasma membrane that then pinches off, and whatever substance has been taken into the cell, that then enters in the vesicle and is taken to where it is needed. Uses of vesicles you need to know, or something like this, for example, where you might have a unicellular organism, might take a food cell you're in, form it into a vesicle, use lysozymes to digest that material, and then use it for whatever the cell wants. Obviously, phagocytosis in disease defense is important as well in white blood cells. It's useful in the placenta because it can again take things into the fetal blood as a result. So there are a couple of key terms that you should just be aware of. Endocytosis is when you engulf materials using vesicles. It can include phagocytosis, which we know is to fight off bacteria and pathogens, and pinocytosis, a term you probably won't use. Actually, technically, phagocytosis means cell eating, pinocytosis means cell drinking. So pinocytosis is the taking of liquids into a cell. And that, therefore, is quite useful for you to see. As we've said before, vesicles are absolutely essential in the division of labour of cells. Vesicles move, and they might take proteins from the rough endoplasmic reticulum to the Golgi apparatus and then out of the cell via exocytosis. So here you've got just a slightly different version of it. So you can see here the stuff might go into the cell via endocytosis, so the plasma membrane is pulled inwards, a droplet of fluid becomes enclosed in the vesicle that is pinched off, and then the vesicle can move through the cytoplasm carrying the contents to where it wants to be. And as we said before, we've got a couple of stages of events in protein synthesis. So proteins are made by ribosomes and they might enter the rough endoplasmic reticulum. Vesicles then take those those proteins, and they might bud them off, take them to the Golgi apparatus. That will then modify the proteins, forming a new vesicle, and then those vesicles will go into the membrane via exocytosis. The vesicles fuse with the membrane, and that then releases this new protein, which might be a hormone, for example, out of the cell into, let's say, the bloodstream. So there's two key terms there, exocytosis and endocytosis. Exocytosis then is when material is removed from the cell, so a secretion of digestive enzymes or hormones, for example, and it's used in lots of different ways, but particular ways mentioned in the specification. Unicellular organisms may use it to expel excess water, so you can see that happening here. Okay, and vesicles surround the water to make contactile vacuoles that then fuse with the plasma membrane, and the water then is released out to prevent the cell from bursting.